All right, here we are with our next macromolecule. We are going to be talking about proteins. So far, we've talked about carbohydrates, we've talked about lipids, and now we're on to our third class of macromolecule, and that is proteins. And if you look at a cell, and if you were to take a cell and get rid of the water, right? If you took a dry mass of the cell, you would find that proteins actually make up half of it. Proteins are a huge part of cells. And they're hugely important for many different functions, including structure of the cell, storage of various molecules, transport of entities around the cell, for communication between cells and between components of living organisms. They're important for movement, and they also are essential for defense against any foreign substances. So we're talking about the immune system there. And there's a great table that you had in your reading, and I have it here on one of these slides. And I recommend that you really study this overview of protein function, because this will help you in answering a lot of the questions on this AP test. However, proteins are tough because they have so many different types of functions. Carbs were kind of like, well, carbs are mostly involved with short-term energy. Lipids are mostly involved in long-term energy with like a few other things. But proteins do many, many things, right? So we have enzymatic proteins, so proteins that are enzymes, and those are really important for accelerating or speeding up the rate of chemical reactions, and we'll talk about what that is. So enzymes is one type of protein. We also have proteins that are involved in structure, right? So we have proteins in our hair, we have proteins in our skin that make our skin kind of nice and smooth. So collagen is important as a connective kind of structural protein in your skin. And as you get older, you make less collagen, and that's why people's skin gets wrinkly, right? And there's a huge market around making people's skin not wrinkly. That's kind of bogus, but we're not going to get into that now. But it's bogus. Right, so they're important for support. They store, right? Amino acids are a form of energy, right? They transport things around cells or around the body. So in your blood, you have hemoglobin that helps to transport gases, in particular oxygen, to where it needs to be. There's hormonal proteins such as insulin that is important in regulating your blood sugar levels. There are proteins that are receptors, so they're proteins that things bind to, and as a result of binding to them, they cause other things to happen, right? So they kind of receive information, also proteins. There are proteins that are involved in movement, right? So your muscles are all proteins. So for contractile and motor functions, we have proteins. Actin and myosin are part of muscles, really important. The cilia and flagella of other organisms, which are these kind of like the hairs or like the tails around like a bacteria that allow it to move, also protein. And of course, we have our defensive proteins, right? So antibodies that help us fight off pathogens that would otherwise cause us problems. All of these functions are performed by proteins. So you can imagine they're hugely important. So we're going to talk about the structure and the properties of proteins. There's a really cool introductory video that I found. I'm not really going to play it for you guys because you can watch it by yourself. I would recommend watching it now. I would recommend going to that link and watching it now. And it's on the website, or it will be if it's not yet. But it's an introduction to how proteins are made. And it talks about the levels of organization of proteins, primary, secondary, tertiary, and it also gives some examples of some proteins. So take a gander at that. Now, going back to our slides, I'm going to kind of go over what's in the video, but I really recommend that you take a look at the video because of the animations. So, proteins are made out of polypeptide chains, right? Poly, again, you hopefully remember, is a word root for many, and peptides is referring to the type of bonds that form between amino acids, right? So we talk about polymers, right? When we talk about these monomers, when we talk about these macromolecules, because polymers are these large molecules that are made up of smaller subunits, right? Are monomers. And in the case of proteins, our monomers are amino acids, right? So a protein is made up of many amino acids that are connected in a chain. You might think of it as like a chain of beads. And there actually only are 20 amino acids that you find in animals. And those 20 amino acids come together in many different ways to form the huge variety of proteins that exist in organisms. There are tens of thousands of different types of proteins that an organism can have. And that's all coming from those 20 amino acids, right? And so here are these 20 amino acids, and they have these wonderful names like glycine, alanine, valine, leucine, isoleucine. And what you'll notice about these amino acids, if you take a good look at them, which I recommend that you do, you don't have to memorize them, but you really should be familiar with the names, just so that you're not thrown off when a question is referring to tryptophan, which is this amino acid right here. What's interesting about amino acids is that they have one part of them that is always kind of the same, right? They have this amino group, they have a hydrogen, and then they have a carboxyl group that's attached to carbon. And then they have something. In the case of glycine, it's just a hydrogen, right? But if we take a look over here at alanine, again, we have our amino group, we have our hydrogen group, we have our carboxyl group, and our carbon. And then we have here a CH3, and that's alanine, right? So what's distinguishing glycine from alanine is the difference in this fourth entity that's attached to our carbon. And this part that differs between amino acids, it's not the same, is referred to as the R group. R for residue. That's really not going to help you, but just remember that. And so these different R groups have different properties. Some of them are nonpolar, right? Some of them are hydrophobic. They don't really like to do things with water. Some of these R groups are polar, right? And so they're good with water. They can be friends with water. They'll dissolve in water. They're okay with it, right? So serine, threonine, cysteine, tyrosine, asparagine, and glutamine are all polar examples or polar amino acids. And then we have other ones that are more acidic, right? They're likely to lose a hydrogen. Then we have ones that are more basic. They're likely to accept a hydrogen ion. And that is all important. These chemical properties of the amino acids is what causes them to do all the things that they do as part of the proteins that they are part of. So let's take a closer look at an amino acid. This is a general structure for an amino acid, right? So this might be a little bit confusing because the ones that you were looking at were kind of one direction, and this is like flipped the other way, but it's still the same thing, right? We still have an amino group, right? NH2. We have a carbon in the middle. There's a hydrogen, and then there's a carboxyl group, right? The COOH. Okay, so we always have this part. That's the same. Any amino acid you look at, it's going to have this. It's going to have an amino group, an NH2 or an NH3 plus. Those two would be the same. Or it has a carboxyl group, right? So it might be a carboxyl group that's COOH, or it could have a COO minus. That's just a question of whether it's in its ion form or not. And then it also always has the extra hydrogen there. Sometimes it's not drawn in, but it's always there. And then it has that one part of it that's different, the residue group, the R group, right? And that varies from amino acid to amino acid, right? Whether you're looking at glycine or alanine, there's a different R group, right? And these amino acids are the monomers that make up proteins. So you can imagine these amino acids attaching to one another in a chain, right? So here we have methionine, glycine, arginine, isoleucine, right? And they are attached to one another and they form a chain. And the chain doesn't just like hang out as a chain. It actually does a very, very interesting and specific folding pattern to become the correct shape of protein, okay? But first let's talk about the bonds between the amino acids. So when these amino acids come together, they react in a dehydration reaction, right? So that means a water molecule is formed as a byproduct, and they form a type of bond that's called a peptide bond. Okay, so the bond between amino acid is a peptide bond. That's why we call them polypeptide chains, right? So when we look at these amino acids, right, they form together in this sort of chain with the peptide bond between them. And when we look at the chain, the sequence, the particular amino acids that are in the chain and the order that they're in is referred to as the primary, or the first, protein structure. So the first thing that proteins do when they are forming is the amino acids will form a chain, right? And a chain will grow. But that's not the end of it. There are 4 steps. So if we look at here on my next slide, this is the levels of protein structures. This is how proteins are made, right? Like I said, first thing, we have our primary structure. That is a sequence of amino acids. What amino acids are there and in what order they are there. The order is important, the amino acids are important. If there's a mistake, if there's a wrong amino acid in the wrong order in the wrong place, bad news. Well, not always, but sometimes really bad news. So first we have our sequence of amino acids. Then those amino acids, so once we have our chain, they will start to coil or fold in different ways, right? And that has to do with the hydrogen bonds, right? So there are hydrogen bonds that form between the amino acids that are on the chain, and as they interact with one another, some parts it will kind of coil up, and some parts it will kind of fold up, right? So that's what's happening during the secondary structure. Coils and folds. The coils are called alpha helices or helixes. Well, helix is singular, helices is plural. So so one of these is an alpha helix. And the fold is referred to beta pleated sheets. And those are kind of folds. And I'll show you some examples of those, I think, on one of my slides. So they start to fold and coil. Then those folds and coils will kind of scrunch up even more, and there's even more kind of interaction between the different R groups on our amino acids, and it kind of forms a very specific sort of shape, right? And that's the tertiary structure. And then the last one is the quaternary, our 4th one, and that's when you have multiple polypeptide chains would come together. So you had, let's say you had one kind of like folded up piece of like a polypeptide chain, and it will like form, a connection with another one here, right? And so it's 2 of these kind of proteins, or these polypeptide chains will come together, and they will together form the protein itself, okay? And I think I have a great diagram for this somewhere. Yeah. So you have this in your readings, so make sure you take a good look at it and study it, and you're comfortable with these levels of organization. They refer to them as levels of organization or levels of protein structure. It's a little bit confusing, but what they're talking about is like the steps that proteins go through to be made, right? So again, primary structure is referring to what particular amino acids you have in what order. Then the secondary structure is when we start to see these beta pleated sheets or these alpha helices forming. Then at tertiary structure, we see these coils and these sheets kind of folding up even more into kind of like not just like a few coils and sheets, but kind of scrunching together even closer in a very specific shape. And then at the quaternary structure, we see multiple polypeptide chains coming together to form one entity, one protein. Okay? So, if we take a look at some examples, what you'll remember, and I've said this before, and this is going to be like a repeating trend, is that shape equals function, right? And the shape is determined by the folding of our polypeptide chain, right? So a protein like collagen, which is really important for structure and connection, right? Collagen is the connective protein in your skin that makes your skin nice and firm and smooth, right? There are these kind of like long proteins that come together and they give tissue structure. If they weren't these long kind of strands, they wouldn't be great at structure. If they were just like a blob, they wouldn't do much, right? On the flip side, we have another type of protein, which is DNA polymerase, which is a type of enzyme, right? And this enzyme is important for DNA synthesis, or the making of DNA. And in order to do that, it needs to have this particular shape where it has this kind of arm here, and there's kind of like another arm here, and the DNA, I think, comes through here, right? Actually, that's not really the best drawing because it really is only one strand. In any case, it needs to have this specific shape in order to make the DNA, right? And if it doesn't have that shape, it's not going to be able to perform that function. And that's really important to know about proteins because there are things that can happen to proteins that make their shape change. And we'll talk about that. So another protein that you will hear about over and over and over again, and you've already heard about a couple of times, is hemoglobin. And hopefully you remember that hemoglobin is the protein that's responsible for carrying oxygen around in blood, right? And the reason why it can carry oxygen around in the blood so well is because it has iron inside of it. And it's a little bit hard to see in this particular diagram, but this is a diagram of hemoglobin. And it shows each of these kind of like spheres is like an amino acid, and it has these four kind of like components to it. It has 1, 2, and there's one in the back, and there's one over here. Those are like the four parts, the four polypeptide chains that come together to make the hemoglobin. And what's cool is that they have these kind of heme groups that have iron in them, and that iron is able to bond with oxygen and carry it to a particular place that it needs to be in the body, right? So it's carried around in the blood. And it's because of this iron that's attached to these polypeptides, this protein, that blood can carry oxygen around. However, if it doesn't have the right shape, it won't be able to carry around the oxygen. The shape allows for the oxygen to bind and then for the protein to carry the oxygen to where it needs to go. However, if you have the wrong chain, if you have the wrong polypeptide, the shape of the molecule changes. So, in a particular type of genetic disease or blood disorder, sickle cell anemia, there is a single amino acid substitution, right? So you have a chain of amino acids and there's one mistake, right? Of the, hundreds or the thousands of amino acids that make up the chain, there is one amino acid that's wrong, in the wrong place, right? And as a result of that, in the case of sickle cell anemia, the wrong protein shape forms. So normally you have a hemoglobin molecule that is this wonderful kind of circular, almost like donut-like shape. And that's really important because, red blood cells have a particular shape to them, right? And in order for that hemoglobin to fit well inside of that red blood cell, it needs to have that particular shape. But when there's an amino acid substitution in the case of sickle cell anemia, the hemoglobin does not form in the same way. It kind of forms these longish strands of protein, and they kind of stick together, so they're not these like nice little separate donuts. They stick together to form these long strands of protein. And that's a problem because then the red blood cell itself that carries the oxygen is the wrong shape, right? So if we take a look at red blood cells, that we have here, a normal red blood cell has this wonderful kind of donut shape to it, right, from the hemoglobin that's inside of it. And that's great because if that red blood cell is kind of traveling through a blood vessel, it just kind of can travel along. It can get squished into half and kind of squeeze through tight blood vessels. Works great. The mutated, the sickle cell anemia red blood cells are these kind of long, fragmented red blood cells. They don't have this donut shape, which you might think is, well, maybe that's not so much of a problem if it's trying to travel through a blood vessel. But actually, they will form kind of these like clumps, right? And they stick together, and they don't allow the blood to kind of pass as smoothly through our blood vessels, and that causes people a lot of pain. It causes them infections, and sickle cell anemia is a really unpleasant blood condition to have. That's unfortunately not very treatable. So here's a photo of normal red blood cells. Right, and you can see again, they have these nice kind of donut shapes to them. And then here you see the sickle cell red blood cells that are mutated. And again, remember, that's simply because there was a single amino acid substitution, right? The wrong amino acid in the wrong place. And so that's why the sequence of amino acids, that primary structure, is so important for the protein to function. Shape and function, those two are connected. A few more things about proteins. So enzymes, we talked about DNA polymerase as a type of enzyme. There's digestive enzymes like salivary amylase, for example, that helps you digest starches in your mouth as you're chewing, right? And enzymes, they are a particular type of protein, and they act as a biological catalyst, right? And the word catalyst, if you haven't heard of it before, if you're not familiar with it, is simply a substance that will speed up the rate of a chemical reaction, right? So they make the chemical reaction happen faster, okay? And the cool thing about enzymes is that they speed up that rate of reaction, but they actually don't get used up themselves, right? They perform a particular function, and then they can be used again and again and again and again, right? So they are reused, they are not used up, okay? And without getting into too much depth about exact types of enzymes, in general, if you think of an enzyme, you can imagine it as a particular type of chemical that has like the ability to hold chemicals together in the correct orientation so that they can react, right? So in the case of here, we have an enzyme that's called sucrase, right? And it's an enzyme that helps break down sucrose, which is a disaccharide, into its respective monosaccharides, glucose and fructose, right? So those are monosaccharides, right? So it breaks the bond between them. So when you have an enzyme, the thing that's being reacted is referred to as the substrate, right? So our substrate in this case is sucrose. Substrate, sucrose will bind to sucrase, which is our enzyme. Whenever you see this ace ending, that means enzyme, okay? Just use it as a hint. Something ace, probably an enzyme, right? So sucrase is our enzyme, sucrose is our substrate. Sucrose binds to sucrase. It allows for a chemical reaction to take place with the help of this enzyme, and now this particular chemical reaction was breaking these two, the bond is broken, and they are released. And now sucrase can go find another sucrose to react with, right? So it's speeding up the rate of reaction. The reaction in this case is the breakdown of sucrose, okay? Enzymes. More about that later. Here I want to just reinforce this idea of structure and function and proteins, right? So proteins have a particular structure, they have a particular shape, and that makes their function, right? Because of their shape, because of, for example, if we go back to our enzyme here, because of the shape of our enzyme, the sucrose will bind to it. If it had a different shape, sucrose would not bind to it. Sucrose wouldn't do anything with it, right? So it needs to have a particular structure for the function to work. And if the structure is disrupted, or if it's broken, then function is disrupted, right? And when we talk about proteins, when we talk about the structure being broken down, being destroyed in some way, we call that denaturation, right? So if you denature proteins, then they become inactive, right? Their function is disrupted. They cannot function. So they've been denatured. And the ways to denature protein, the most common one is temperature. If you heat proteins up, you denature them, right? You cook some proteins, you are denaturing them. They will no longer function. But other ways you can change them or denature them is pH as well as the concentration of any salts. Okay, so keep those in mind, that the ways that you can denature proteins is with pH, concentration of any salts, and temperature. And if you take a gander here, here's an example to show you kind of what this denaturation process is, right? So you have a normal protein here. It has a specific shape, and then you heat it, let's say, or you put it in some acid, and suddenly the shape of it is broken down. The heat has caused the bonds to break, and now it's like a totally different shape, right? And so it no longer functions. In some cases, there can be renaturation, but that, for the most part, does not happen. Denaturation happens, and proteins are done so. So if you actually have noticed, when you cook an egg, when you first put the egg on the frying pan, it's clear, right? It's kind of like a transparent or a translucent substance. And as you heat it, it turns white, right? The color kind of changes. And that's actually as a result of the fact that there are proteins in the egg that are being denatured from the heat. Okay? So that's always really cool to think about. Let's do some practice questions. So, first of all, when we think about protein structure, the hydrogen bonds are really important in stabilizing or assisting the formation of the protein. And I didn't talk about this in a lot of depth, so this question is actually a little bit tricky, but give it a go. Try your best. And I'm going to go with the answers now, right? So we talked about the primary structure, right? The primary structure of a protein is a chain of amino acids, right? Oops. And the secondary structure is when that chain starts to fold into alpha helices and beta sheets, right? And then the tertiary structure is when those sheets and helices fold up even more, and the quaternary structure is when you have multiple polypeptide chains coming together, right? And so anytime your protein is folding and kind of like attaching to other polypeptides, it's hydrogen bonding is happening, right? Because all of this is happening in water. So it turns out that the secondary, the tertiary, and the quaternary structure are all using hydrogen bonding in some way to stabilize or assist the protein structure, but not primary structure. Primary structure is simply the sequence of amino acids, and what's most important there are the peptide bonds, not the hydrogen bonds. So for that reason, E is your answer. If we go on to our next question, this one's kind of connecting the things that we've learned about so far. So if we think about polysaccharides, if we think about triacylglycerides, which is also sometimes called triglycerides, those are the same. So we're talking about lipids here. And if we look at proteins, what things are similar about them? Okay, so read through your options, don't get tricked by them, and choose the best one. So I'm going to start from the bottom again. So polysaccharides, triacylglycerides, and proteins, do they all contain nitrogen? And the answer to that is no, right? Triacylglycerides don't have any nitrogen in them. Okay, are they decomposed into their subunits by dehydration reactions, right? Remember we said dehydration is actually involved in the formation or the synthesis of polymers. So that one we know is not true. Are they all synthesized from peptide bonds forming between monomers? That's also not true because peptide bonds are only formed between proteins, right? And are they synthesized from dehydration reactions? And that turns out is true, right? So when proteins are forming and the peptide bond is forming between amino acids, water molecules are formed, right? In polysaccharides, when they come together and they form that glycosidic bond, again, a water molecule is formed. And we didn't really talk about it in too much depth with the lipids, but also when you take your glycerol and your fatty acid and they form a bond between them, then water is also formed, okay? But just for good measure, let's look at option A. Are they all synthesized from monomers in a process of hydrolysis? And you know that's not true because triacylglycerides don't have monomers in the same way that polysaccharides and proteins do, and also hydrolysis is used for breaking them down and not forming them. Okay, so your answer there is B. And that's all I really want to talk about for proteins. I'll see you for nucleic acids next.